Good afternoon. I am here with you again to present another presentation by the Virginia State University Spring 2016 Cytology Group. I am Anthony Fields along with my colleagues, and we will be presenting to you today a paper by the name of microRNA 34A and a regulation on mesenchymal cell proliferation via the platelet-derived growth factor receptor beta RAS MAP kinase signaling pathway.
Hi, I'm Shantara Stewart, and I'll be going over the RAS MAPK signaling pathway. So first, you start off with the epidermal growth factor, which is the EGF, as you look at the picture to the right. That will bind to the epidermal growth factor receptor, which is the EGFR, which is a receptor tyrosine kinase, which will activate the tyrosine kinase activity. The growth factor receptor will then phosphorylate the tyrosine residues, and the growth factor receptor bound protein 2, which is GRB2, is the adapter protein that will bind to the phosphorylated tyrosine residues of the activated receptor. The SOS, which is a protein, it's the SUN 7 list, binds to the RAS, which is a membrane-bound protein, causing the SOS to activate. The inactive RAS will bind to the GDP, which is a nucleotide, and the SOS promotes the removal of the GDP. RAS binds to the GTP and becomes active. Then the RAS will bind to the beta RAF, which is the effector protein, which then phosphorylates and activates MEK1 and 2. And MEK is the mitogen activated protein kinase. MEK will phosphorylate and activate the ERK1 and 2, which is the MAPK kinase, the mitogen activated protein kinase. Then activates Jun and FOS, which are transcription factors. And then Jun and FOS will form a heterodimer and bind to the motif of the DNA. That will lead to expression of genes in the growth factor, and cell proliferation is activated. So how this relates to our paper that we're discussing is the MIR34, which is the microRNA 34, which is the growth factor, will bind to the platelet-derived growth factor receptor beta, and that will bind to the platelet-derived growth factor protein, which that will then, the bindage of those two will activate the RAS MAPK signaling pathway, which also downstream will activate target genes as far as cyclin D1 and E, which will control the cell proliferation.
Good morning, my name is Bertrita Raditsis, and I will introduce the paper for you. So we'll start with mesangial proliferative glomerulitis. It's a disease of the kidneys that affects the glomeruli. Its key characteristic is the proliferation of mesangial cells, and is also a prominent feature in glomerular disease. And these include two types of nephropathy, and that is immunoglobulin nephropathy, which is inflammatory, and non-immunoglobulin nephropathy, which is non-inflammatory. In each case, hyperplasia is exhibited in all or most of the glomerular mesangial cells, along with several levels of extracellular matrix accumulation. Reducing and inhibiting the proliferation of mesangial cells is important in treating proliferating glomerular diseases. The experiment used a model called the anti-THY1 model, which is a standard animal model for mesangial proliferative glomerulonephritis. Includes THY1 antibody and THY1 antigen, and that is together THY 1.1, and that is found in rats. It is also detected with the clone OX7. And the mechanisms of mesangial proliferative glomerulophoresis. Glomerular damage occurs when the macromolecular antigen antibody complexes are formed in or on site. They become deposited in the mesangial area. This leads to both mesangial cell proliferation and inflammation and stimulus of activation. Once this happens, shrinkage and mesangial cell proliferation occurs. Consequently, a variety of inflammatory mediators and ECM components are released. These events subsequently exacerbate inflammation. This results in further damage to the molecular and barrier of the glomerular filtration membrane along with albuminuria. MicroRNAs are a single-strand RNA molecule. They are 21 to 25 nucleotides. In eukaryotes, microRNAs are post-transcriptional regulators. MicroRNAs do not code for proteins, but microRNAs regulate proteins by inhibiting translation or degrading target mRNA. This is made possible by its ability to bind to a specific target site. MicroRNA is involved in many processes and is also associated with many diseases in humans, including renal diseases. MicroRNA 34A is a component of that and is found to be associated with tumor proliferation of many cancers through target gene regulation. And the hypothesis. The purpose of the study was to clarify the role of microRNA 34A in renal mesangial cell proliferation.
Okay, since we saw the RAS-MAP kinase pathway, we know that growth factors are RTK cell surface receptors that will activate RAS by hydrolyzing GTP and is bound to GTP in the active state. So the purpose of the siRNA or silence RNA is because it interferes with gene expression by causing mRNA to be broken down after transcription. So that means there is no translation. So the target gene and that they're examining are the silencing of the platelet-derived growth factor receptor beta. Here you see the sense and antisense strands. So the sense strand is the 5 to 3' end, and the antisense is the 3 to 5' end. And in this paper, they will be referring to it as the SI-COM.
In Table 2, we're going to talk about the real-time quantitative PCR primers for these listed target genes. The purpose was to look at the transcript levels for each of the genes, and there you see there's a sense, and that's the coding strand that runs from 5' to 3', and the antisense is the complementary strand that run from 3' to 5', and it's showing the base pair fragments. And then in Table 3, this is the PCR primers for the 3' untranslated region fragments for the target genes of the PDGFR beta and the cyclin-dependent kinase 2 and cyclin E. These are the target genes that were amplified from the rat mesangial cells from the PCR, and each fragment contains the target sequence for the growth factor of the MIR34A. So right here we're targeting the antisense, the cDNA, because that's the complementary strand.
Ki67 is a protein that is exclusively associated with cell proliferation. Changes in the expression of Ki67 were found in the THY1. They used a periodic Sheriff stain in figure 1A, and N is the control. And you can see on day 3 that it started to exhibit Ki67. Day 5, it increased. Day 7, there was a peak. Day 10, it started to decline, and in day 14, it went back to normal levels. And figure 1B, they used a detection, they detected the Ki-67 by immunochemistry. Compared to the control N, higher levels were found on each time point in THY nephritis. This shows that the cell cycle remained active between 3 and 10 days, increasing on days 3 to 5, peaking on day 7, and declining on day 14, 10 to 14. They use a labeling index to calculate for the labeling index calculated for the Ki-67 positive cells versus the glomerular cells on days 3, 5, 7, 10, and 14. And as you can see, N, which stands for which is in place of the control, was at normal levels. Day 3, with the experimental rats, they saw it increase. Day 5, it increased further. Day 7, it peaked. Day 10, it began to decline. And day 14, it stabilized. For figure 2, the microRNA 34A detection, they detected microRNA 34A gene expression by real-time qPCR on days 3, 5, 7, 10, and 14. And of course, you have the control on the front. MicroRNA 34A was found to begin to gradually decrease with the progress of pathological processes. On day 3, which was the lowest point, on day 3, it began to decline. And 7, it declined. 5, it declined further. On day 7, it was at its lowest point and began to normalize again on day 10 and day 14. Both microRNA 34 and pathological processes normalized on day 14.
My name is Loretta Granum. In this figure, the researchers aim to determine the effects that microRNA 34A has on the proliferation rate of mesangial cells in the cell cycle. Here we look at figure 3A, which shows us the optical density of renal mesangial cells over the course of 72 hours. If we look here, we see the normal control and the negative control of the microRNA 34A bear similar results, giving us the first indication that microRNA 34A does play a regulatory role in the proliferation of these mesangial cells. After 24 hours here, there is a reduction, and right here, in the presence of microRNA 34A mimics, after 48 hours, we notice a slightly significant reduction. And it's even further reduced, even more significant reduction at 72 hours in renal mesangial cell proliferation. The results show us that microRNA 34A mimics are influential to the proliferation suppression of these cells. Now in figure B, we're looking at the results from a flow cytometry method, which focuses on the cellular DNA content. If we look at the normal and control results, we'll see that there's somewhat a small range in the DNA content. So looking down here, in an elevated number of cells, if you look up here, the microRNA 34A mimics, on the other hand, tend to have an extended amount of DNA content, indicating an extension of the G0 and G1 phases, which is consistent with the cells being restricted at the cell arrest or cell growth phase. And we see a reduction in the cell number, meaning that the G2 or mitosis phases are also reduced. Table 4 shows us further support of the results indicated in figure 3. By looking at the percentage values for the microRNA 34A mimics, there is an increase in mesangial cells that are in the G0 or G1 phase. So that's here in the highlighted region. which is consistent with the extension of these phases shown in the previous figure. There is a significant reduction in the percentage of mesangial cells in the S phase, being that the cells are unable to commit to this particular phase due to being arrested at the restriction point. And lastly, the G2 or mitosis phase further supports the results in figure 3 because we see even less of a percentage of mesangial cells at this point. Being that cells are not able to proceed past the restriction point in the presence of microRNA 34A mimics to undergo the S phase or the G2 or mitosis phases, growth factors are not binding to their receptors to stimulate cell proliferation.
Here is an examination of Table 5. Table 5 shows the relationship of microRNA 34A and that it targets the beta-derived growth factor, the platelet-derived growth factor beta in the 3' untranslated region. Complementary binding indicates that microRNA 34A can inhibit the beta-derived growth factor receptor.
Figure 4. Through dual luciferase assays on fireflies and ranellas, we know that microRNA 34A have binding sites at the 3' untranslated regions of platelet-derived growth factor receptor betas, making it a target gene of microRNA 34A. In order to understand microRNA 34A's effect on the target gene's expression, dual luciferase plasmids were used to monitor the changes in expression of the target gene fused to a reporter vector, which is SideCheck2. This formed a SideCheck platelet-derived growth factor receptor beta plasmid, which was co-transfected with either a microRNA 34A mimic or a microRNA negative control into the renal mesangial cells. In those co-transfected with the mimic, there's a significant downregulation in the firefly luciferase activity. Figure B shows the results of real-time qPCR to show that platelet-derived growth factor receptor beta mRNA is expressed in renal mesangial cells following transfection with either microRNA 34A mimics, the mRNA negative control or the normal control. In Figure C, Western blot analysis was conducted to observe the protein levels of platelet-derived growth factor beta and the phosphoplatelet-derived growth factor beta, which is the activated form, after transfection. We can see here that there's a significant increase in the expression of platelet-derived growth factor receptor beta in the phosphorylated form in the renal mesangial cells, which were transfected with microRNA 34A mimics, compared to the normal and control group and negative control groups here. Being that expression was reduced, researchers were able to determine that the microRNA 34A likely targets the inhibition of platelet-derived growth factor receptors. In Figure D down here, Western blots were used again to reveal protein expression of platelet-derived growth factor beta and phosphoplatelet-derived growth factor beta, receptor beta, in the anti-Thy1 nephritis rat models. Based on the results, day 7 shows the most pathological change, if you look here. And it is on that day that the expression of phosphorylated platelet-derived growth factor receptor is significantly greater, meaning a growth factor is bound. This indicates that platelet-derived growth factor receptor is a key regulator in the ability of anti-Thy nephritis to proliferate. For Figure 5, as we know, the cell cycle requires the assistance of proteins to regulate the progression of the cell cycle. Flow cytometry showed us that microRNA 34A mainly impacts the G0 or G1 phase. Therefore, this study focuses on the expression of the G0, G1 phase regulatory proteins and their expression levels. Here in Figure A, researchers measured the expression levels of the mRNA for cyclin D1, cyclin E, in cyclin-dependent kinases 2, 4, and 6, and P27. The results show a significant reduction in the microRNA expression of cyclin E in the cells transfected with microRNA 34A. Cyclin E is one of the cyclin proteins needed for the progression into the S phase of the cell cycle by binding to cyclin-dependent kinase 2. Also, in the microRNA 34A mimic cells, there is a significant increase in P27, which is a cell cycle protein kinase inhibitor. Figure B shows us the expression levels of the same cell cycle proteins. At this point, we see a significant reduction in the expression of all proteins except the P27 inhibitor. So here we look at the reduction in expression under those transfected with microRNA 34A mimics. The results are consistent with what the researchers hypothesized, which was microRNA 34A targets the G0, G1 cell cycle proteins. And here you see increased activity for P27. There were previous studies conducted to support the idea that cyclin D1 and cyclin-dependent kinases 4 and 6, which form their own complex, are target genes. Therefore, in figure C and D, the focus is placed on cyclin E and cyclin-dependent kinase 2. These are the two proteins that showed major reductions in mRNA expression levels when co-transfected with microRNA 34A mimics. So once again, luciferase activity is measured by cloning the 3' untranslated regions of both SideCheck2 of proteins into SideCheck2 to form the plasmids SideCheck cyclin E and SideCheck cyclin-dependent kinase 2. Renal mesangial cells were co-transfected with microRNA 34A mimics in the plasmids to determine whether or not microRNA 34A is a downregulator of cyclin E and cyclin-dependent kinase 2. The results reveal that there's a significant decrease in firefly activity, therefore microRNA targets these proteins.
Okay, and figure 6 for A, this is a Western blot to the left that is showing the expression of the proteins in the conditions of normal, which are regular cells. Con, the miRNA negative control, and mimics, which is a transinfected with the MIR34A mesangial cells. So as you see for RAF1 and ERK1 and 2, here, they show no change, but MEEK1 shows a significant decrease. And you can see it over in this bar graph. In the normal cells, all the levels seem to be even in the bar graph, but as far as the con cells, they show a vary in the levels, but not much for the proteins. And KRAS, phosphorylated RAF1, phosphorylated MEEK1, and phosphorylated ERK1 and 2 seem to be downregulated, as you can look in the Western blot. The mimics have a major difference in expression levels, which indicate that for every phosphorylated protein, the levels were low, meaning that they were activated. But the significant protein level that we're looking at is the phosphorylated MEEK1, because it barely has any expression right here, indicating that when phosphorylated MEEK1 is phosphorylated and activated, the MIR34A is inhibiting the expression. Also, they're pointing out that MEEK1 is the target gene when it is being phosphorylated. In part B, they're showing the days right here for the proteins being expressed. And for day 7, it's showing a distinct visibility that the phosphorylated MEEK1 has increased levels of the protein with relation to expression, but it's not as high for the phosphorylated MEEK1 on day 14. And you can see that right here in the bar graphs.
In figure 7, researchers wanted to confirm the effect of microRNA 34A on the platelet-derived growth factor receptor by introducing small interfering platelet-derived growth factor receptors on the cell cycle, its related proteins, and the Ras-MAPK signaling pathway to measure its influence. The purpose of small interfering RNA is that it's a tool used in RNA interference techniques to promote gene silencing and targeting of the microRNA for degradation. Here we see that renal mesangial cells transfected with small interfering protein derived growth factor, platelet derived growth factor receptor beta, reduce cell proliferation hours, similar to the effects that microRNA 34A mimics produce. In figure B, flow cytometry is used again to indicate that small interfering platelet-derived growth factor receptor beta also extends the G0, G1 phase and reduces the G2 or mitosis phases here in comparison to the normal and the negative control. Here in figure C, Western blot analysis shows a significant decrease in the cell cycle regulatory proteins, as we saw with the microRNA 34A mimic effects. So here we'll see reduction in the proteins, the cell cycle regulatory proteins, and an increase in the P27 inhibitor protein. The results also show a decrease in the protein expression of the Ras-MAPK signaling pathway proteins, KRAS here, the phosphorylated RAF1, phosphorylated MEK1, and phosphorylated ERK1 and 2. If we look here on the bar graph, RAF, MEK, and ERK1 and 2 did not show a change, which provides evidence that the small interfering platelet-derived factor receptor beta and microRNA 34A have similar effects of suppressing the platelet-derived growth factor receptor and the suppression of the cell cycle protein cyclin D1 and E, and the cyclin-dependent kinases 2, 4, and 6, as well as promoting a significant increase in P27 expression. We can understand that the small interfering RNA and the microRNA 34A directly inhibit the platelet-derived growth factor beta. Then it impacts the Ras or MAPK pathway, followed by the cell cycle regulatory proteins. Table 6 shows us further support of the results indicated in figure 7 by looking at the percentage values for the small interfering platelet-derived growth factor receptor beta. There is an increase in mesangial cells that are in the G0, G1 phase, consistent with the extension of these phases shown in the previous figure. And we see a significant reduction in the S phase of mesangial cells, being that they're unable to commit to the particular phase, being that they're stuck at the restriction point. And lastly, the G2 or mitosis further supports the results in figure 7 because we see less of a percentage of mesangial cells at this point. Due to the fact that the cells are not able to proceed past this restriction point to undergo S phase or the G2 or mitosis phases, growth factors are not binding to their receptors to stimulate cell proliferation.
Okay, now we'll take a look at figure 8. So we already established that microRNA can inhibit normal cells. So here that we examine the rat mesangial cells and exogenously stimulated cell proliferation with FBS, which is fetal bovine serum. And we had found that even with the stimulation of the fetal bovine serum, that there was still a attenuation in the amount of cells that would actually proliferate in comparison to the controls. We saw at the 10%, there was a slight increase, but at a higher concentration, there still was an inhibition of cell proliferation. And in figure 8B, we also look at the flow cytometry, and it shows that there is a clear reduction in cell proliferation in comparison to the control of the 20% of the SBS and the 20% FBF with the mimic. So that we also conclude, even with cell stimulation, that microRNA 34A reduces the amount of cells. As we look at figure 8C, we see that with the mimic in comparison to our control, that there is a reduction in the amount of proteins that are produced during the PGFR and Ras/MAP kinase signaling pathway. So as we see that you have the constant stimulation in the Western blot with the bold lines. If you look over to the mimic, you see that the bands get slightly smaller. So meaning that the proteins that are there are not fully being fully expressed. And here as well, you see the protein levels, expression levels as well, in comparison to the mimic and our controls. You can see that the mimics have a great reduction in the amount of cyclin proteins and Ras proteins that are supposed to induce cell proliferation.
Table 7, it shows the percentage of mesangial cells in G0, G1S, and G2 in phases. They use exogenous factors, a fetal bovine serum at 20% to stimulate mesangial proliferation to see what microRNA 34 alpha would do with proliferation. We already know that microRNA 34A can inhibit the target gene, platelet-derived growth factor beta function of rat meningeal cells by suppressing GO/D1 phase. They found that higher concentrations of serum accelerated cell proliferation. That was 20% of the fetal bovine serum. And with microRNA 34A at high concentrations, along with that microRNA 34A, the concentration, the cell proliferation rate went back to normal, even with the high levels of serum, and that was for 10% serum culture.
Okay, well, finally, just recap everything that we have examined. We understand that microRNA 34 inhibits the platelet-derived growth factor beta, and it also has the ability to inhibit MEEK, which is a downstream, phosphorylated MEEK1, which is the downstream activator of the Ras/MAP kinase pathway. So overall, we basically can conclude that the microRNA 34A regulates mesangial cell proliferation by directly inhibiting the PDGFR beta and MEEK in the Ras/MAP kinase pathway. And it influences the GO and G1 phase by extending these phases and reducing the S phase and the G2 mitotic phase. The microRNA 34A directly targets CDK2 and cyclin E to extend the GO phase, causing cells to extend the mitotic phase. So this would be a good way to target future strategies for tumor reduction. And this will conclude our presentation. And thank you, and see you next time.